Most people think arterial plaque works like a blocked drain. Cholesterol builds up, the pipe narrows, eventually it clogs. That picture is wrong, and understanding why it is wrong is the difference between managing this disease and actually reversing it. Plaque is not grease coating the inside of a pipe. It is an active inflammatory process, driven by your immune system, influenced directly by what you eat, and in many cases, reversible through specific dietary compounds that most people have never been told about. Today, I am going to show you exactly what those compounds are, which foods contain them, and how to combine them into one meal that targets the actual biology of plaque, not just the numbers on a lab report. Stay with me through the end, because the last ingredient on this list is the one almost nobody talks about, and it may be the most important of all. What plaque actually is. Before we discuss the meal, you need to understand what arterial plaque actually is at the biological level. Because once you understand the mechanism, you will immediately see why certain foods work and why the standard advice often falls short. Here is what happens inside your artery wall. Your innermost arterial layer, called the endothelium, is a single layer of cells that forms a protective barrier between your bloodstream and the deeper vessel tissue. When these cells are healthy, they produce a molecule called nitric oxide, which keeps your vessels relaxed, flexible, and resistant to damage. But when the endothelium becomes damaged through high blood pressure, chronic inflammation, or blood sugar instability, it develops microscopic gaps. LDL cholesterol particles slip through those gaps and become trapped in the arterial wall. Here is the critical step most people never hear. That trapped LDL does not immediately cause harm. What causes harm is what happens next. It oxidizes. It goes rancid, the way cooking oil goes rancid when exposed to heat and air. And oxidized LDL triggers your immune system like an alarm. Your immune system sends specialized cells called macrophages to clean up the damage. These cells consume the oxidized LDL, but they cannot process the volume efficiently. They become overwhelmed, engorged with cholesterol, and transform into what researchers call foam cells. Foam cells accumulate in clusters inside your arterial wall. That accumulation is plaque. Here is what makes this dangerous. Those foam cells are not static. They are inflamed, fragile, and eventually they rupture, releasing their contents and triggering the blood clot formation that causes heart attacks and strokes. And the plaques most likely to rupture are not always the largest ones. A plaque that is only 40% obstructive can rupture and kill because the danger is not the size. It is the degree of inflammation inside. Understanding this changes everything about how you approach the problem. The solution: reverse cholesterol transport. So the real question is, can the cholesterol trapped inside those foam cells be extracted and removed from the artery? The answer is yes. Your body has a built-in mechanism to do exactly this. It is called reverse cholesterol transport. HDL cholesterol, the kind you want more of, functions as a collection vehicle. When an HDL particle encounters a foam cell, it docks on the cell's surface and activates a protein called ABCA1. This protein acts like an ejection pump. It pulls cholesterol out of the foam cell, loads it onto the HDL particle, and that particle carries the cholesterol back to the liver for elimination. The foam cell shrinks. The plaque regresses. The disease reverses. The problem for most people is that this process is running at a fraction of its capacity. ABCA1 is underactivated. HDL is not functioning efficiently. And the result is that plaque continues building even in people doing many things right. But ABCA1 can be activated through food. Specific compounds in specific foods switch this pathway on. And that is exactly what this meal is designed to do. Ingredient number 1, arugula. The foundation of this meal is arugula, 2 to 3 cups, raw. Arugula is one of the richest dietary sources of nitrates, and nitrates, through a pathway your body converts them through, produce nitric oxide, the molecule your endothelium needs to stay functional. Here's how it works. When you eat nitrate-rich foods, your salivary glands concentrate those nitrates in your saliva. Beneficial bacteria on your tongue convert them into nitrite. When you swallow, gastric acid converts nitrite into nitric oxide, which enters your bloodstream and signals your blood vessels to relax and widen. Rigorous clinical trials have demonstrated that dietary nitrates measurably improve endothelial function, lower blood pressure, and reduce arterial stiffness within hours of consumption. Your vessels physically open up. But here is something critically important. This entire pathway depends on the beneficial bacteria in your mouth, and one common habit destroys them completely: antiseptic mouthwash. Studies have confirmed that regular mouthwash use abolishes the nitrate to nitrite conversion, completely eliminating the nitric oxide benefit from dietary nitrates. If you use mouthwash daily, the arugula stops working for this purpose. Brush thoroughly, floss regularly. That is sufficient. Do not use antiseptic mouthwash if you want this meal to do its job. Eat the arugula raw. Cooking degrades the nitrates significantly. Ingredient number 2, high phenolic extra virgin olive oil. The second ingredient is high phenolic extra virgin olive oil, 3 to 4 tablespoons, used to dress the salad. Olive oil contains a phenolic compound called oleocanthal. It is what creates that peppery, slightly burning sensation at the back of your throat when you swallow quality olive oil straight. And that compound does 2 specific things for your arteries. First, it is a powerful anti-inflammatory, inhibiting the same enzymes targeted by ibuprofen, reducing the inflammatory activity inside arterial plaque and stabilizing it. Second, and this is the critical mechanism, oleocanthal directly activates ABCA1 expression. It switches on the gene that produces the cholesterol ejection pump in your foam cells. Research conducted on macrophage cells treated with oleocanthal showed dramatic increases in ABCA1 activity and measurable cholesterol efflux, meaning the cholesterol was physically being pumped out. The PREDIMED trial, a landmark study involving 7,000 high cardiovascular risk participants over 5 years, found that a Mediterranean diet supplemented with extra virgin olive oil at this quantity produced a 30% reduction in cardiovascular events compared to a low-fat diet group. Not a small effect. And the mechanism tracked directly to the phenolic compounds. The quality of the oil matters enormously. Cheap, refined olive oil has been stripped of its phenolics. You need cold-pressed, single-estate, recently harvested oil, ideally from a dark glass bottle with a harvest date within the past year. When you taste it straight, it should burn the back of your throat. That burn is oleocanthal. No burn means no therapeutic effect. Do not reduce the oil to save calories. The oleocanthal is the mechanism. Removing it means eating a salad, not activating reverse cholesterol transport. Ingredient number 3, pomegranate arils. The third ingredient is 1 cup of fresh pomegranate arils, the seed sacs, added directly to the salad. Pomegranate contains a specific class of polyphenols called punicalagins. These compounds have a documented and unusual property. They bind directly to LDL particles, coating them and making them resistant to oxidation. Remember, oxidized LDL is what triggers the entire foam cell cascade. If LDL cannot oxidize, macrophages do not consume it, foam cells do not form, and plaque does not develop. A clinical trial that administered pomegranate juice to participants for 1 year measured carotid intima media thickness, a direct measure of plaque burden. The plaque thickness decreased. The investigated mechanism? LDL oxidation had dropped dramatically. The LDL particles were physically protected by the punicalagin coating. Pomegranate also increases the activity of an enzyme called paraoxinase, which lives on HDL particles and breaks down oxidized lipids. Your HDL becomes more functional, more efficient at its job of clearing cholesterol from the arterial wall. Use the whole fruit arils, not juice. Juice is high in sugar, which introduces its own inflammatory burden. 1 cup of arils gives you a clinically meaningful dose of punicalagins with minimal sugar impact. Ingredient number 4, vitamin K2 source. The 4th ingredient addresses something almost never discussed in mainstream cardiovascular conversations: arterial calcification. As plaque matures, calcium deposits begin forming inside it. The arterial wall stiffens. Vessels lose flexibility. Blood flow becomes increasingly impaired. And a high coronary artery calcium score, measured by CT scan, is one of the strongest independent predictors of cardiovascular events. Here is the mechanism behind calcium regulation that most people have never heard of. Your arterial smooth muscle cells produce a protein called matrix GLA protein, MGP. Its biological job is to prevent calcium from depositing in the wrong places, specifically to keep calcium in your bones and out of your arterial walls. But MGP only functions when it is activated, and it is activated exclusively by vitamin K2. Without adequate K2, MGP remains inactive. Calcium migrates into arterial tissue. Your vessels calcify. With adequate K2, MGP is activated, calcium is redirected to bone, and existing calcification can begin to reverse. Published research from the Netherlands demonstrated that long-term K2 intake from dietary sources was associated with significantly reduced arterial calcification and lower cardiovascular mortality. Supplementation studies using a specific form called MK7 at 180 micrograms daily showed that calcium scores stabilized or decreased in participants. K2 is found in fermented foods, particularly natto, a Japanese fermented soybean preparation that contains the highest known concentration of MK7. It is also found in aged cheeses such as Gouda. If neither is practical, an MK7 supplement at 180 micrograms taken with this meal achieves the same biological effect. This is the only nutritional compound with published human evidence for reversing arterial calcification. It belongs in this protocol. Here is the complete meal assembled. Start with 2 to 3 cups of fresh, raw arugula as the base. Add 1 cup of pomegranate arils. Dress generously with 3 to 4 tablespoons of high phenolic extra virgin olive oil. Add a small amount of sea salt and black pepper if desired. On the side, either 50 grams of natto, 3 ounces of aged Gouda, or an MK7 supplement of 180 micrograms taken with the first bite. Eat it slowly. Chew thoroughly. The nitrate to nitrite conversion begins in your mouth. The bacteria on your tongue need adequate contact time to do their work. Consume this meal once daily, consistently. Consistency is what produces measurable results. The biological processes we are activating, ABCA1 upregulation, MGP activation, LDL oxidation resistance, are cumulative. They build over weeks and months. Eat it at the same time each day. Lunch works particularly well as nitric oxide levels will peak during your active afternoon hours. If you want objective evidence that this is working, here are the measurements that matter. A coronary artery calcium score, obtainable through a CT scan, gives you a baseline number and a way to track change over 18 to 24 months. A stable or declining score is evidence of regression. An advanced lipid panel, beyond just total cholesterol and LDL, includes LDL particle number, particle size, HDL particle number, and apolipoprotein B. These markers reveal what is happening at the level of actual plaque risk, not just the numbers most doctors currently measure. High-sensitivity C-reactive protein measures systemic inflammation. You want this below 1.0 milligrams per liter. Elevated levels indicate ongoing arterial inflammation that dietary changes may not be fully addressing. These tests give you data. They take the guesswork out and tell you clearly whether the protocol is producing the biological changes it is designed to produce. A few things I want to be clear about. This meal is not a replacement for prescribed medication. If your physician has prescribed a statin or other cardiovascular medication, continue taking it as directed. Dietary intervention and medication work through different and often complementary pathways. Discuss any changes with your doctor. This meal is also not a one-time fix. The research on saffron and lutein for eye health shows results at 3 to 6 months. The arterial biology here is similar. Consistent daily practice over months is what produces measurable change. Occasional use produces none. And this meal works best as part of a broader dietary pattern, one that also reduces processed foods, refined carbohydrates, and seed oils. The salad is the foundation, but what surrounds it matters too. Arterial plaque is not a verdict. It is a biological process, and biological processes respond to consistent, informed inputs. The 4 ingredients in this meal target 4 distinct mechanisms: nitric oxide restoration, ABCA1 activation, LDL oxidation prevention, and calcium regulation. Together, they address the actual cellular biology of the disease, not just one marker on a blood test. You now understand more about how plaque forms and how it can be reversed than most people ever learn. Use that understanding. Apply it consistently. If this was useful, share it with someone you care about: a spouse, a sibling, a friend managing cardiovascular risk. The right information at the right time genuinely changes outcomes. And if you want more honest, evidence-based conversations about health after 60, subscribe to this channel. We are here every week. Take care of your arteries, and I will see you in the next one.